Hi, everybody, and welcome back to Biology Bios. My name is Jim Edelman. I'm an associate professor in the Department of Biological Sciences at the University of Memphis, and my purpose with this podcast is really twofold. First, I want to let folks know that most Thursdays during the semester, we have talks by visiting researchers at 4 p.m. in Ellington Hall. And that's folks from all over the country and world talking about the really cool biological research that they do. The second purpose is to have those folks here learn a little bit more about them, how they got where they are, and what really excites them about the research they do. In addition, I'd like to say a quick disclaimer that I borrowed these questions from Rhett Miller's excellent podcast called Wheels Off, so please check that out as well. But without further ado, let's get to this week's guest. Thanks so much for being here. Dr. Steve Nowicki, welcome to Biology Bios. Thanks so much for being here.
Delighted to join you.
So I generally start by asking folks if they would give a brief intro of who you are, where you are, the sort of broad brushstrokes of your research interests.
Yeah, my name is Steve Nowicki. I'm a faculty member at Duke University in Durham, North Carolina. I've been here for well over 3 decades, and during that time, mostly interested in studying the function and evolution of natural signaling systems, or what you might call animal communication. My lab and I have worked on a variety of organisms, including even some work on humans. But the vast majority of our work has been done with birds and bird song, partly because it's just a great model system where we can manipulate signals, we can record signals, we can work with animals both in the lab and in the field, and ask a lot of interesting questions to get at how that signaling system works and what we can learn from that about signaling systems in general, including our own language.
So, I'm going to, my last interview was with Scott Sakaluk from Illinois State, and he suggested a question that I haven't been asking people, but you kind of drove the bus this direction. So I'm going to ask you, for folks who, aren't themselves scientists, and at the risk of being blunt, what is the broader value of studying songbird signaling? And you hinted at human language there, but can you go into that a little more?
Yeah, I'm going to answer in sort of a practical sense, but then I'm going to, if you allow me, try to wax eloquent about, sort of the broader issues of aesthetics and appreciating nature. So maybe surprisingly, birds singing, and especially how they learn to sing, has long been an important experimental model system for understanding aspects of human speech learning and development. Now, that's a little surprising because, the sparrow singing in your tree doesn't have much to say compared to the youngster learning how to speak in your house. But there are remarkable parallels that were actually first really elucidated way back in the 60s and 70s by a scientist named Peter Marler, who showed that, like human speech, songbirds, not all birds, but this group of birds called songbirds, need to listen to adult models when they're young in order to copy those songs and eventually produce them themselves. Just like young human babies need to listen to the adults around them to eventually learn how to produce speech.
Sure.
In terms of the developmental process, phenomenologically, we know that human speech development is gradual, that young children are listening early on before they're saying anything. And then as they begin to produce speech themselves, say, as a 1.5 to 2, maybe 2.5-year-old, they gradually put it together, producing what the speech people call babbling. And the babbling gets to be more and more complete until finally the child is making words and then putting words into sentences. Mm-hmm. So too, the same is true with birdsong. Again, it's not quite as complicated what they end up with in terms of the function of this, but the young bird has a period of time early in life when it has to hear these songs. That's what we might call a sensitive period or a critical period. And then it starts practicing them. And it does what we call subsong, which is very unstructured, the same way babbling is. And it goes even further, right? Because now, as people have looked over the last few decades in the brain mechanisms, it turns out there are remarkable parallels in terms of things like the lateralization of speech is compared to lateralization of the control of birdsong. Aspects of the areas that are involved in the brain actually, now bird brains and human brains are structured hugely differently. But if you now, with modern molecular techniques, kind of trace their evolutionary similarities, you can find that there are parallels there as well. So the last thing I'll say about the practical aspect is that one interesting thing about bird song that's a little different from human speech is that humans can continue to learn throughout their lives. It's hard. I mean, if you start learning a language as an adult, it's harder than if you learn it as a 1 or 2 year old. And you may always have a very accented version of that language. Mm-hmm. You may never quite nail it. With birds, for many species of birds, actually, I would say most songbirds, they actually have a critical period that closes down. And, for a song sparrow, that is about at the 2 to 3 month old age. And after that, they can't learn anything new. For other birds, though, they continue to be able to learn. And so the reason that that's been interesting to neuroscientists is that it gets at this issue of adaptive plasticity in brains, right? So what it means is that there are some aspects of bird brains, probably on a molecular level, that allow some species to stay open and continue to learn and others not to. So that gets to sort of fundamental questions about the neural molecular genetic mechanisms underlying plasticity, learning. But it also has attracted attention because it has potential therapeutic implications.
Sure.
So another very famous scientist who was one of the real founders of the neural study of birdsong, a person named Fernando Nottebohm, actually wrote a paper decades ago that was a summary of what he had been learning about ongoing plasticity, especially through neurogenesis in birds. And it was a plenary paper for a big conference, but it had it its subtitle, "A Hope for a New Neurology." And his argument is, if we can understand what enables or disables plasticity in a bird brain, think of what we might be able to do in terms of therapies for humans who have suffered a brain injury or stroke or something. So that's a long answer to why we get funding from places like the National Institutes of Health to study this. For me personally, I'm delighted that that's the case. But for me, and I think for maybe a lot of people, understanding animal signaling kind of connects me to understanding music, right? I can listen to a Beethoven string quartet and have the emotion of that wash over me. I can also analytically take apart that string quartet and say, how did Beethoven do that? I might do the same thing with Tom York and Radiohead. How did that sound, affect me?
Yeah.
What is it that he did technically? And I think that that's sort of, to me, something that's interesting about studying animal signaling is that it is the music of the world around us. And we can appreciate that music and thus appreciate the world we live in more by understanding a little bit about what those birds are doing and how they're doing it.
Yeah, I also, as someone who studies sort of, basic science, just trying to figure out how the world works, like that definitely resonates. Also, as somebody who likes to play music, everything you said there, I'm totally on board. That's awesome. So you've worked on mostly sparrows, but you've worked on other critters, you've worked on humans. And I know that your students have done projects that are, outside of your particular interests as well. And I'm curious right now, of the projects that you have going, is there one that you're particularly excited about? And why does that one light you up?
Oh, wow. Well, I just started a collaboration with a new colleague here at Duke, Sarah Lipschitz,
Cool.
who is interested in, sort of neurogenomics of behavior, and also happens to work on birds as model systems. Um, so there's sort of two reasons this lights me up, right? So we have long known that birds recognize differences, geographic differences, basically dialects. And sometimes it's pretty obvious to we humans what the birds are cueing in on, right? You know, you can
Mhm.
hear a dialect of white-crowned sparrow from one valley or the next valley out in California, and even without being a practiced, birder, you can hear that they sound different. But there are some species that tell the difference between populations, and we don't have a clue what they're listening to. We
Mm,
just can't. And an example of that would be song sparrows. So song
yeah.
sparrows are widely distributed across the East Coast and up through Canada and down through the Sierras, down through California. Um, they have very complicated songs. Every song type is a complicated thing. A male might have, 6 to a dozen different song types. In a population, there could be hundreds of song types. So if you look at the variation between two populations, it's huge and it's so complicated. We don't have a stepping stone to sort of say what are the birds actually listening to.
Hmm.
So, and we've tried and we failed to find things. So, what we have done recently is start to look at um, uh, genomic expression in response to the playback of local versus foreign songs to sort of take it into the brain and to see if we can find basically what you'd call transcriptomic differences.
Yeah.
So that's exciting, not because it's working yet. I mean, we actually have started that and it is working enough to get us excited about something coming out of it. But it's exciting because it opens up a new tool for
Hmm.
getting at the question of what are those birds really interested in. Coupled with that, with the advances of artificial intelligence tools, we can now start asking AI, all
Yeah.
right, you tell us what you find that are different, and then we can go test it in the field. Um, so coming back to what's really been an old question, I mean, literally, I mean, this has been something that we've worried about for 20 years. Um, with new tools, it has been quite exciting.
Yeah, I think that AI, I was going to ask about that as well, because that seems like a sort of training algorithm sort of thing. Like, here's a bunch of Washington song sparrows. Tell me why they're different, or pick them out from the ones from New York or whatever. But I think there's another cool thing there that you have super multivariate data, both in the song and now in the transcriptomics. I mean,
Yeah.
it's got like AI written all over it, categorizing those transcriptomic responses too.
Yeah, yeah. Well, it is true, and I have to say that uh, I am learning a ton, right? Sarah is the molecular biologist, and I am her student, even though I am her elder colleague, by 30 years. But it's been really super fun to, not only, go and do those techniques in the field because we're doing this stuff out in the field. So, we have to kind of take the dry ice and all the stuff
Oh
out in the
man.
field and the other thing that's exciting about that to Sarah and to me is that that kind of work that Sarah has done, that people like Kim Rosval at Indiana are doing, tends to be terminal, right? Because
Yeah.
you need to get the brain tissue, right? You need to, and
Hmm.
you're not going to get that as a sample. So the other thing that Sarah and I are doing, and this is also exciting for a different reason, is we're asking how much transcriptomic information can we get out of blood?
Oh, cool.
So birds have nucleated red blood cells, so there's a fair amount of DNA in there, and um, and the early results that we've got is that we do see differences in the blood, and that opens up again a whole new set of kinds of experiments you can do because you can test the same bird
Yeah.
repeatedly because you don't have to kill
Yeah.
it. You can just take literally 100 microliters of blood, which is not a problem at all for the bird, and let the bird go. So we
Yeah.
did all of that work in the field, and the birds, they weren't happy about being caught and having a drop of blood taken, but they flew off and were happy
Yeah.
to still be, and stayed around, and we could catch them again. So we were able to
Compared to
do
the
this
alternative.
compared to the alternative. Yeah.
That's super cool. Yeah, I think we're excited about trying to get some proteomics going as well because I feel like, working in these non-model systems, so not race mats, race mats, mice, rats, and humans. Um, it's hard to have all the tools, but I think we're maybe at this really exciting time. And yeah, transcriptomics is one of those, but proteomics as well. Like we're at this exciting time where we might be able to kind of leapfrog with this new technology and kind of get past some of the specific molecular diagnostic things and get to these big global questions. Yeah, it would be great if we had those tools as well, but like, yeah, I think you're going to be able to do really cool stuff with that.
So you use the word leapfrog, and that is exactly the right word. And I'm thinking by analogy of the development of telecommunications in developing countries,
Absolutely.
right? Where in what we would call developed countries, we went through the telegraph, telephone lines, all of that, and now finally we've gotten to our cell phones and being able to do banking online. Whereas in developing countries, they've just leapfrogged that. They don't need those telephone lines the same
Yeah.
way that we might not need some of those older techniques now that doing transcriptomics or proteomics has become, frankly, cheaper
Yeah.
and faster. I mean, I remember, I sound old, but I remember the old days when people like you were in intro bio and I was enthusing about how we might finally get the whole human, genome sequenced. And
Yeah.
now it's like, hey, here's another human question. Let's just sequence a dozen of these guys, and we'll have the data for you on Tuesday.
Yeah, yeah, it's bonkers. It's absolutely bonkers to see that happen. Um, okay, so I wanna take a little bit of a pivot and talk about less about the actual research itself and more about your path to getting here. So was there a time that you knew you wanted to be a scientist, or was it kind of a gradual, slow burn? How did you end up where you are?
Um, it was like uh, it was like a lightning bolt. Um,
Okay.
so the high school I went to was just a public high school, not great, not bad. It didn't have anything in the way of science teaching that was all that good, and biology was only taught in ninth grade. There was no AP biology or any upper level biology courses, environmental science, or any of that. And with apologies to Mr. Freer, the ninth grade biology course was kind of dull to me because it was mostly dissecting frogs and field
Sure.
pigs, and I I didn't know why we were doing it, and I wasn't very good at it. So I sort of left that, and the other science courses that I had to take were not all that exciting. Whereas the high school did have a really good music um, program. I mean, good for that high school, and I got really excited about music. And so when I went to college, my goal was to be a music major. Um, and I ended up achieving that goal, but I went to a liberal arts college where you couldn't just study one thing,
Hmm.
like with most liberal arts curricula, you had to have some distribution of courses, including courses in the sciences. And so I had to take some science courses, and I had a couple of buddies as a first year student who had taken the intro bio course at this place, and they loved it. And they said, "Hey, you should take this course." It was co-taught in the right way. Both professors were there at every lecture. They
Huh.
they you know they would go back and forth. They would comment on each other's lectures, and they were different. One was a molecular biologist, a cell and molecular biologist who worked on these really interesting, single-celled organisms called chlamydomonas, and the other was an organismal biologist who actually happened to work on animal behavior. They had a great relationship. I took that course and it blew me away. It was like so interesting. And it
Huh.
was interesting because, you know, there were problems to solve and you could solve them. Not all of them easily, but it was
Mm-hmm.
like, it wasn't as hard as trying to solve, fundamental theories in physics. And it wasn't as abstract as, trying to solve questions in philosophy. It was tangible
Hmm.
and it was beautiful. I mean, it struck me as just being beautiful, and so I took that course and I said, all right, I've got to be a biology major. So that was the lightning strike. And then I took more biology courses and I just really liked it. And I liked being in college. And so I said, oh, I'm going to go and get a PhD in biology. Now, that wasn't very sophisticated, to be quite honest. And, it's not what I would recommend to many students today, but that's what happened to me. And the rest is history.
That's fantastic. So I've got to say that I took intro bio from Steve way back in the day. Um,
That
and
ages me.
it ages both of us, man. Um, but I was considering whether I was going to be a music major or a bio major. And I was also taking, I took all the theories with Jairo Moreno. I don't know if you ever met him at Duke, but he's a fantastic jazz bass player and like just brilliant dude. And I was sitting there just like, which way do I go in my major? And I ended up being a bio major, music minor, but I think it was some similar logic along those lines. And you mentioned the course, and I'm also teaching intro bio right now. So there's this potential of those courses where you can just show how cool is this thing? And I remember thinking that a bunch of times in your class, being like, that is really cool. We think of these big classes as just like, okay, well, you got to get through intro bio to get to the interesting stuff. It was still one of my favorite classes in college. Like you can do them right. And Steve, I'm preaching to the choir here because you put a lot of thought into that.
I spent a lot of time in that course, and that was actually me paying it forward because intro bio in when I was in college changed my life. I was a grad student at Cornell, where a very famous introductory biology program existed, by a person named Bill Keaton, who wrote an extremely influential textbook in the 1960s that was really a dominant textbook through the 1980s. And I TA'd his course, and it was really, really well taught, very thoughtful. And it was a huge course. I mean, at Cornell, there were,
Sure.
probably 600 kids who would take it. Um, and then I got to Duke, and the introductory biology course wasn't that good when I got here. I mean, with apologies to the people who were teaching it, but they were sort of doing it out of a sense of duty, and it was team taught, but they didn't coordinate very
Yeah.
well. And I would hear students who were my advisees saying, I'm a biology major in spite of
Ooh.
introductory biology, and it really bothered me. Um, so I went to the chair of the department as an assistant professor. I was teaching a neuroscience course at the time, but I was ready to do some other stuff, and I said, let me take over the introductory biology course. And he said, no. He said, get tenure first. So then I got tenure a year or so later. And then I came back and I said, okay, now can I? And he said, yes. And, I didn't have to do that. I was teaching other courses that were very popular and that I enjoyed teaching, but I felt like it was really important for a place like Duke to have an introductory biology program that was attractive to students, that would get them excited about it. And I taught that for a number of years, and then passed the torch on to others. And so I spent a lot of time. That's when my hair started going gray, but it was certainly worth it.
I've, I've noticed a few more gray hairs this semester now that you mention it. But it's so cool to get to learn that stuff again. Stuff either that, maybe I didn't learn the first time or like that it's just been so long that I haven't thought about. It's just it's a way to see biology anew again and just be like, damn, that is cool.
Yeah, yeah.
Um, okay, so a couple more here along this line. So in science, we have a lot of external critics. We have reviewers. We have, people, naysayers in general. But we also put up some internal criticisms as we think, oh, maybe that idea isn't that great. Maybe I shouldn't pursue that. And I'm wondering what your experience has been with that and how you either silence that voice or maybe make it work for you, or I don't know, maybe you don't have that voice, which awesome.
Um, I do have a voice that is more of, to be honest, a um, sort of general self-doubt as to whether I should be here at all. This is imposter syndrome.
Yeah.
And you'd think at this point in my career, I'd get over it. But I do every once in a while think, gosh, how lucky I've been. But in terms of this, I worrying about having wrong ideas, imposter syndrome can lead you to think, oh, the idea can't be that good.
Right.
But my good fortune, and it is luck, is to have encountered a few collaborators along the way who I can, well, I mean, trust is one word I would use, who don't necessarily take me seriously. So I have a lot of wacky ideas.
Okay.
And I can share those wacky ideas. And I have had collaborators, my longstanding collaborator, now retired, Bill Searcy at the University of Miami, being an important one. Another one being my longtime collaborator, Susan Peters, who I married before we started collaborating, who can, hear the 10 ideas and say 9 of them are wacky. They're just stupid. But it enables, and I encourage people to do that. Find people who are not going to be, personally critical, who are going to be critical in the right sense of the word, and sort of say that idea doesn't make sense, or maybe that idea makes sense, but we need to rethink this. Multiple minds are always better than one. And if you can find, other people who you feel comfortable with and can share your ideas with and not feel challenged when they tell you that the idea is bad, because they're not saying you're bad, they're just saying that idea is not good, that I think moves things forward. And I frankly will say that this is another reason why it's so important to have diverse voices, right? Because if, I only talk to people like me, we're going to share a lot of ideas. But if I talk to people who have different lived experiences than me, it just increases the possibility that together we're going to be able to find, the best way to move forward. And together we're going to be able to, sort of create new ways of thinking about something that would otherwise be really impossible or very hard to do. I really think that in spite of what some people think, diverse perspectives are essential for excellence. It's diversity is excellence, but you have to have the communication, and that is finding people with whom you can just sort of feel very comfortable saying, hey, here's an idea, and very comfortable having them say, I don't understand that, or that doesn't make sense, or that's a wacky idea.
But yeah, I like your point though, that a certain group could just say, well, that's a wacky idea. But if you have a diverse group, there might be somebody in there that's like, it is wacky, but what if?
Exactly.
And you tweak it, and then all of a sudden, wacky idea becomes super cool.
Yep. Totally.
That's awesome. All right. So I think you've hinted at this with that answer, but I think I'm going to ask this other question as well, because there might be other things here, which is thinking of a undergrad version of yourself in today's world of biology. So you meet the 20-year-old Steve Nowicki in today's world. What advice would you give that person about moving forward in science and biology?
That's a really good question. I'm not sure that I know how I would answer that really, but I would say that as a younger person, what as I developed through my career, as I grew older, I grew more comfortable with criticism. I think young people are susceptible. They want to be liked, right? I have a big nose. When I was a young person, it really bothered me that I had a big nose. Now I'm thinking, well, it's just who I am, whatever. And I think that, I would have told my 20-year-old self to loosen up sooner, because it took me a while to get comfortable and to loosen up. And like I said, I was very lucky along the way to find people with whom that was easier done. And now I think I could do it with anybody. And I actually actually, again, going with my new collaboration with my new, colleague who literally just started her job here a year and a half ago. It's a lot easier for me now to feel comfortable with her telling me that I don't know what I'm doing, or, I mean, not in a mean way, but in a nice way, to sort of take instruction from this person who, I know that has ways of looking at things which are different, that are different from mine, and maybe better, maybe worse, but certainly different. And, it would have been hard for me as a 20-year-old to have that same kind of relationship with somebody who's really coming at things from a very different perspective. So I, tell my younger self to just loosen up. Don't take yourself too seriously.
Yeah, that's good advice. And I like how you keep bringing it back to that community. I think the older I get in science, the more clear it is to me how collaborative and how a good community I've had collaborations that are not as good as others, right? And finding those people is really important. Finding that community is really important to doing good work.
I would add another thing, which is something I also tell my students at every level. To be generous is part of that, right? I mean, so in academia, ideas are, our currency, and we have ideas. Um, and we have to share those ideas to get them better, right? That's part of the community. And there, I think, a tendency, and maybe I had this tendency when I was younger, to say, hey, this is an idea and it's mine, and I've got to keep it. And it's understandable, right? Because there is a sense of competition in academia. We can't deny that. And if you share ideas, you do stand a chance of, so-called getting scooped, and that has happened to me where, but on average, and I can now average it over, 3 decades or more, it pays to be generous, because the number of times that it backfires and you get bitten by it is, that's no fun, but on average, it's better to be generous because you make more progress that way, and just trust people. I prefer to trust people at the risk of occasionally being disappointed, as
Yeah.
opposed to not trust people and then never getting disappointed.
Yeah. Yeah, I like that. I like that a lot. Like intellectual goods shared or not diminished.
Yeah.
Yeah. Um, all right, so let's take it out of science for a second. And what do you do when you're not sciencing?
Um, Cook
Oh,
is one. I still do a lot with music, although the performance side of it has gone down. But your listeners are going to now say, all right, so this guy is completely off the wall. But it turns out that during the pandemic, just before, by chance, our son, and I bought my wife, my also longtime collaborator, an accordion.
oh, nice.
Now, my wife is a really good musician too, and she actually plays a lot of kinds of instruments, and she's really interested in various kinds of folk music and roots music. And she was playing hammered dulcimer. She's very good at keyboards. And but as a young person, long before I knew her, I didn't meet my wife until we were in our early 30s, but long before, when she was in high school, she actually played accordion. And she still had, when I met her, people can hardly imagine this, but high schools used to have accordions along with their trombones or whatever. And she had one.
Hmm.
And we carried it around for, a couple of decades. And it eventually, the bellows rotted away.
Hmm.
And so, not really as a joke, but because my wife likes music and she likes new challenges, our son and I bought her an accordion for her birthday, which is in early March of 2020.
Oh,
And then the pandemic hit. And she found a great teacher, a local performer who's really great, who could give lessons on on Zoom. And so, as we were all locked down, Susan was really taking off with the accordion. And now she plays accordion. She plays in a band called Scandemonium that plays, Scandinavian
oh,
folk music.
wow.
Yeah, no, it's really a fun band. But so one of my retirement dreams is to become a busker. So I own a 3/4 B flat tuba, and I play lower brass instruments. And so my goal is to, brush off the tuba when I retire. And I think it'd be a great act, right? An accordion and a
Accordion
tuba.
and a tuba together at last. I love it.
You would put $1 in that hat, I'm sure.
I 100% would. Oh my gosh. I want to see this. That's your retirement plan, that you two on the road busking different cities. Like,
Right.
that sounds fantastic. I love it. Um, okay, so my last question I'm going to throw right back at you and ask, what else what have I not asked that you wish I had asked you?
Well, you've asked a lot of really great questions. Um,
And to be fair, I asked this question because somebody suggested that I asked it, like Scott had suggested that other question. So I just keep intellectual good
yeah,
shared and not
right.
diminished, right? I just keep stealing other people's questions.
So here's a question. I'm not sure I wished you'd asked it, but that one could ask is, where do your ideas come from?
Ooh, that's a cool question. Yeah. And what's the answer?
Um, my ideas largely come from keeping my eyes open and just noticing stuff. Some of that is keeping my eyes open in terms of trying to really remain broad in what I know about science and the reading I might do. Some of it literally is keeping my eyes and ears open as I'm walking out in nature. Um, uh, and one notices something, and that thing that you notice may or may not turn into a well-formed question. And that question then might or might not turn into something that could be a hypothesis that might actually be in an area that you feel is important to understand. Um, but it's basically just keeping a really broad um, set of interests and keeping your eyes and your ears and all of your senses open to the world around you. And I think that works on all levels, right? I mean, so as somebody who works on the level of organisms and is, working out in the field as much as working in the lab, we can visualize that. We can imagine walking through the woods and seeing something and saying, hey, what's that about? But I think it would work on the molecular level. I think it works on any level of organization from, the planetary sphere down to, biomolecules, to just ask yourself, what's different here? What
Hmm.
do I notice about this system that, and then sometimes you go and you look at it and you say, oh, well, people have worked on that, in which case you might find ideas that stem from that. And sometimes you might say nobody's even asked about that.
Yeah.
um, maybe in my uh, work in the last few years, it's the question of how um, categories in the way we perceive the world actually affect the way we might think about how signals evolve. Um, uh, Understanding that we and other organisms are often perceiving the world in categories that our brains are forming. They're not really there. Our brains are making stuff up or putting things in boxes. And if we go, for example, and measure the color of the variation, the color of a bird from orange to red, and we know that that's a carotenoid-based color, and we know that the carotenoids are involved in a cost-based signaling system that will affect mate choice and so on. If we assume implicitly that the bird is seeing those colors in a continuous fashion, just as we're measuring them, then we might miss something.
Yeah.
But if instead we say, hey, we see orange or red, right? When you look at a rainbow, there's categories there which aren't real. Those are our brains making it up. And so then you have to ask the question, well, so is the animal, does a bird see rainbows? And if it does or doesn't, is going to change the way you think about how signaling systems might evolve. So that's an example of just sort of noticing something and coming up with a question that might turn into a hypothesis that you might be able to come up with predictions that are testable for.
So when you have those ideas, do you carry a notebook? Do you just say, "Huh, I wonder"? Do you little voice memo on your phone or something like how? Because I feel like that's part of this process is maybe you get 100 of those, and like you were
Yeah.
saying, you have colleagues to be like, "Well, 99 of those are wacky."
All of the above. All of the above. I mean, I didn't have voice memos on a phone back when I started out, but a little notebook. My handwriting is awful, so I might write something down, listening to a talk in a meeting or something
Hmm.
like that, and hardly be able to decipher it. That example I just gave of thinking about categorical perception of color, that came out of a journal club discussion that, I said, um, hey, this paper is assuming this. How do we
Hmm.
know that's true? And then a postdoc working with me in the lab, a woman named Eleanor Caves, now at university, said, that's a good question. We should test that, which is interesting because I'd say, I have no clue how to test that. And Eleanor said, oh, I think we could figure that out. And, by the end of that evening, we had an emerging hypothesis, and it was just a matter of sort of blurting something out in the context with somebody like Eleanor. And Patrick Green was another person there, also a faculty member at Brown, where, after a half-hour conversation, you've sort of taken a thought and turned it into what might be a testable idea. And actually, in that case, 18 months later, it turns into a Nature paper.
Yeah.
That was a if you don't mind the basketball analogy, that was a full court shot that landed. It doesn't always work that way, but that one did.
Yeah, that's super cool. I really like that. That's man, yeah. How do you get your ideas? That's a great question. Awesome. Well, Steve, it has been an absolute pleasure. I'm gonna let you get back because I know you have some travel coming up, as well as travel here, and we'll have your talk next week on the 20th of March. So I'm very much looking forward to that. But yeah, thank you so much for doing this. Really appreciate it.
Oh, my pleasure, Jim. And I look forward to visiting you and your colleagues out there in Memphis.
Awesome. See you soon. Thanks so much to our guest, Dr. Steve Nowicki. You actually have two chances to see him talk next week. That's Thursday, March 20th at 4 p.m. in Ellington Hall on University of Memphis campus, and Friday, March 21st at the Memphis Chess Club for Nerd Night. Hope to see you all there, and thanks so much for being here.